Biochemistry has come a long way in the past few decades, sometimes achieving what has been thought impossible. But have you ever imagined a world in which science could bring the dinosaurs back to life? Well, that's what the movie Jurassic Park is all about. The iconic movie was directed by Steven Spielberg in 1991 and stands as a cinema classic, garnering fame across the globe. However, Jurassic Park is more than just a blockbuster hit. It's a story with some very real science behind it. Steven Spielberg explores the ambitious creation of a theme park featuring genetically cloned dinosaurs and blending advanced genetic engineering with the recreation of an extinct species. The plot delves into the ethical and scientific dilemma of cloning, with a particular emphasis on the unpredictability of experimentation. As the park's security system fails and the dinosaurs escape, the movie leads into a thrilling struggle to survive. It combines special and practical effects to serve as a cautionary tale of using genetic engineering.
Life will not be contained. Life breaks free, it expands to new territories, and it crashes through barriers painfully, maybe even dangerously, but, there it is.
Central to the plot is the character John Hammond, whose ambitious vision was to make a theme park that offered an unprecedented experience to bring dinosaurs back to life and allow people to see them from a controlled environment, a mix of scientific curiosity and commercial incentive.
But let's talk about the science of Jurassic Park. Early in the movie, the scientists discovered dinosaur DNA that was extracted from mosquitoes, which fed on the dinosaurs millions of years ago. These mosquitoes were found to be preserved after being trapped in amber, which kept the DNA somewhat intact. However, this DNA was incomplete, so they filled in the gaps using DNA from a frog, which in the real world doesn't make much sense, as frogs and dinosaurs are pretty distinct from one another. However, this is a movie after all. After the DNA is made, the scientists were then able to create viable dinosaur embryos by inserting them into ostrich or emu eggs to develop. The eggs were incubated and then hatched, which resulted in live dinosaurs that were then put into their respective prehistoric habitats to be raised. But wait, how much of Jurassic Park is even accurate? Is it even possible to create a Jurassic Park from the science in the movie? The information mentioned in the movie does create an interesting story, but it's not scientifically accurate. One point of contention is the idea of using frog DNA to fill in the gaps in the fragmented dinosaur DNA. In order to fill in the gaps, one would actually need to know where the gaps are. For this to happen, scientists would need the whole genome sequence of the dinosaurs they want to bring back. Additionally, using reptilian or avian DNA would be more preferable if you did have the whole genome. They're more closely related to the dinosaurs. If scientists were able to find viable dinosaur DNA, then cloning might actually be a possibility. Unfortunately, however, this has not been done yet, because DNA is vulnerable to the elements. Even if the DNA sample was intact, contamination from modern DNA and RNA would be a problem. Moreover, the oldest DNA found to date is about 1 to 2 million years old. Dinosaurs existed 66 million years ago, so it's pretty unlikely that any dinosaur DNA is even still around, or even stable, which makes Jurassic Park pretty improbable. However, DNA is remarkably capable of self-preservation, and the secrets locked within bone fossils do reveal an interesting story. Under optimal natural conditions, DNA can survive just over 1 million years. Fossilization, however, has a toll, subjecting DNA to partial degradation and chemical modifications. The method of how ancient DNA withstands the test of time adds even more challenges to the analysis. While the extraction of DNA samples is relatively straightforward, the true challenge lies in sequencing, given the substantial degradation. Nature's role in preservation becomes evident where DNA located in the permafrost boasts a sequencing success rate of 62 to 78%, compared to 23 to 67% in moderate climates and a mere 2 to 4% in hot, arid regions. Beyond climatic factors, preservation hinges on an interaction of chemical processes including pH, oxidizing reducing potential, irradiation, and the chemical composition of bone and soil. Hydrology also plays a crucial role, yet the exploration of these dynamics remains largely unstudied. In a groundbreaking endeavor, a team in China unveiled an extraordinary preservation of cartilage cells from a 125 million year old dinosaur called a Caudipteryx. The cell's incredible conservation are due to a fine layer of volcanic ash, entombing the dinosaur down to the cellular level. Silification further mineralized the cells, providing an unparalleled glimpse into the past, with an uncommonly well-preserved nucleus containing original biomolecules and the threads of chromatin. However, despite advancements in amplifying ancient DNA through PCR, the sequencing process still presents challenges. The resulting sequences from ancient DNA are often incomplete or contaminated by microbial DNA. But pushing the boundaries of exploration, the oldest successfully sequenced DNA hails from a mammoth and dates back a staggering 1.6 million years. But now let's take a journey through the fascinating world of cloning technology. Picture this. It's the 1970s and scientists are just beginning to clone frogs. Fast forward a couple decades and voila, in 1997, we witnessed a groundbreaking moment. The cloning of Dolly the sheep, the very first mammal cloned from adult cells. Now how do they do it? The technique they used is known as somatic cell nuclear transfer. Imagine a sheep egg cell, and scientists carefully removing its nucleus. Then they take a nucleus from a mammary gland cell of Dolly's mother and delicately insert it into the egg cell. This egg now holds a DNA blueprint identical to Dolly's mother. Next, this egg is implanted into a surrogate mother. And there, in the nurturing environment of the surrogate, the egg grows until the birth of Dolly. Fascinating, isn't it? And guess what? This very process is still the go-to method for cloning of other mammals today. Cloning can also be done through the process of embryo splitting. This is done through in vitro fertilization, where an egg is fertilized by a sperm cell and then implanted into the uterus. Once division starts, the embryo is split into two sets of two cells, which results in identical embryos. This is the same process that occurs naturally to result in twins. But there are many limitations that may cause issues to arise and make this cloning process a little more difficult. There are a few examples of long-term genetic issues that have arisen in cloned animals. For example, Dolly the sheep was the first mammal to be cloned, and she developed a lung disease that scientists think was due to the premature aging of the mammal due to being cloned from an adult cell. Scientists are unsure of the long-term health effects of cloned animals, which can set them back because they can't gather enough data on the cloned animal before it just dies. Genetic diversity can also be lost if a large number of animals are just cloned, and therefore are very similar in genetic makeup. This can damage ecosystems and have the opposite effect intended. But let's get back to bringing animals back. A lot has changed since 1991, and our understanding of genetics and cloning has advanced significantly. Yet the question remains in how much of Jurassic Park was pure fiction and how much is actually rooted in reality. Can we do something that could achieve results not for dinosaurs but perhaps other species? Have we developed new technologies? Are there more ethical concerns to be aware of? So many questions can be answered just from a single thought. The term de-extinction was coined in 2012 to describe the reversal of animal extinction. But research on this topic existed as early as the 1920s. The viability of the field increased exponentially upon the creation of the San Diego Frozen Zoo in 1972, where DNA from thousands of extinct and nearly extinct animals are stored. De-extinction initiatives have gained recent public attention, centered around the woolly mammoth, dodo bird, and the northern white rhino. The woolly mammoth revival project, which has been spearheaded by the biotech company Colossal Biosciences, has the ambitious goal of bringing back the woolly mammoth from extinction by 2027. The dodo bird de-extinction project, also by Colossal Biosciences, has a similar goal of reviving the recently extinct dodo bird. While other projects which are focused on conservation and saving endangered species, like the Northern White Rhino Preservation Project and the DNA Bank for at-risk animals, are also investigating animal cloning as a solution. Although the thought of reviving extinct animals is exciting, there are still ethical considerations that need to be taken into account. As mentioned by Dr. Ian Malcolm in Jurassic Park,
Yeah, but your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should.
many scientists argue that the money spent on reviving extinct animals would be better spent elsewhere to prevent existing species from going extinct in the first place. There's also an argument that if the government was responsible for caring for these resurrected species, all the current conservation efforts would lose funding, resulting in an overall decrease in biodiversity as roughly 2 species would go extinct for every 1 that could be revived. If private funding was allocated to existing conservation program for living species rather than de-extinction efforts, we would see a large increase in biodiversity as roughly 2 to 8 times more species would be saved from extinction. We also don't have any clear notion of how to reintroduce long extinct animals into their natural ecosystem. Due to climate change and deforestation, the environments where these extinct creatures like woolly mammoths once lived have changed drastically. Introducing these animals back into the wild could wreak havoc on a fragile ecosystem and deplete native populations. This focus on de-extinction also promotes the idea that extinction is no longer scary. The public mindset becomes, deforest? No problem, we can reforest. If we drive an animal extinct, that's fine, we can de-extinct it. Spending millions of dollars to revive a few species will not compensate for the thousands of species that have been lost due to human activity. But then this raises the question, are we reviving these species for our own enjoyment? Or is it because it's what's best for the planet? Whatever the answer may be, there you have it. A look into biochemistry, cloning, and the wild concept of de-extinction. From the imaginative world of Jurassic Park to the cutting-edge laboratories of today, we've seen how far science has come and how far it might go. But as we stand at this crossroads of scientific possibility and ethical responsibility, we're reminded of the delicate balance between what we can do and what we should do. Whether it's bringing back the woolly mammoth or preserving the species we still have, the future of our planet's biodiversity hangs in a delicate balance. As we continue to push the boundaries of science, let's not forget our role as caretakers of the Earth, ensuring that our scientific endeavors improve rather than diminish the natural world around us. Thank you for watching our video on the intersection of science, ethics, and the future of biochemistry. We hope you learned something today and answered any questions you didn't even know you had. And if you haven't seen the movie Jurassic Park yet, we highly recommend it. It's a classic.